All right. So my name is Osama Atalla. I'm a postdoc at Dr. Verchot's lab, Texas A&M University, and my talk today will be on the potential host range for rose virus. So first, roses are among the most valuable ornamental plants due to their highly economic value, which brings about $10 million a year to the industry. The genus Rosa includes about 200 species with 18,000 different cultivars or maybe more. So the rose rosette disease is among the most devastating threats to roses in North America, as it causes between 5 to 10 million US dollars lost annually. The disease symptoms include, as you can see here in those pictures, the hyperthorning, witch's broom, compact canopy, and reddish discoloration of the newly emerged leaves and the shoot. So there's many symptoms, and list goes on and on. So our research was established to see whether rose rosette virus is present throughout the infected bushes and what other plants would host the rose rosette virus when grown next to the infected roses. And can we mimic the RRV sab inoculum by using our infectious clone of RRV on different host plants, and how long it will take for the RRV to establish an infection, a systemic infection, I mean, and whether or not the RRV can cause symptoms on other hosts. So first things first, so the inoculums that we have made is being driven from RRV infected Julia Child and Duscher cultivars that's been confirmed RRV positive by RT-PCR using RRV specific primers. And those plants were used to prepare SAB as well. And we prepared the inoculum by grinding 1 gram of tissue in 10 mL of phosphate buffer supplemented with Silwet as a surfactant. And then for the detection of the RRV in our experiments, we used RT-PCR and we tested 4 previously published sets of primers that anneal to either the viral nucleocapsid or the potential movement protein of the virus. And we found that the primer bed developed by DiBello in 2018 was the most sensitive to our samples. So we decided to use it throughout our studies. So first, we wanted to see whether RRV occupies the whole infected plant at once. So we started collecting symptomatic and non-symptomatic tissue from multiple sites. As you can see here in this picture on the left, the sites 1 and 2 are symptomatic, while the sites 3, 4, and 5 are not. And then we ran them into the RT-PCR after total RNA extraction, and as a result, we detected the RRV nucleocapsid exclusively in the symptomatic tissue at this point, while the healthy looking tissue was free from the virus, as shown by the RT-PCR. Just a second, let me find my pointer real quick. So look that the virus isn't evenly distributed or it's not evenly presented in the infected shrubs or bushes. So next, we wanted to see if RRV can invade some other plants, some other host plants that are not shrubs or not bushes as roses. So we decided, we selected like some herbaceous plants representing families of the Cucurbitaceae, Solanaceae, Leguminaceae, Malvaceae, Amanitaceae, and Brassicaceae. And we tested those plants as potential hosts for RRV by mechanical inoculation with the RRV SAB that we previously extracted. And those plants like cucumber, squash, pumpkin, pepper, tomato, tobacco, peas, spinach, and Arabidopsis. So after 3 weeks of inoculation, we started collecting tissue and analyzing the samples by RT-PCR, and we found that most of those plants came up positive for RRV, except for Arabidopsis and Lubini. So we also saw some other plants that were not as sensitive as the others to the virus, like yellow squash and Quinopodia quinoa, as they show only 1 or 2 infected plants. So we've looked at the process or the progression of the RRV in infected plants and those infected plants on a weekly basis. And we saw that the RRV takes somewhere between 2 to 5 weeks to establish a infection in herbaceous plants. Also, the RRV titer or infection may decline or disappear after pre-infected, from these pre-infected plants within like few weeks of inoculation. As you see here, And this red box indicates that those plants have lost the infection after 5 weeks of inoculation, while it was present before. So, as for symptoms, inoculated Quinobodia quinoa or amaranth colored spinach, cucumber, and datura showed some minor leaf symptoms like leaf deformation, as you can see here in this picture, indicated by arrows, and leaf chlorosis. But most of these plants, didn't show nothing remarkable or distinguishing. So, and lastly, we wanted to test if our RRV infectious clone is as infectious as the contagious SAP that we've used before. So we combined those 7 RRV segments of the infectious clone together that we previously cloned into a binary vector, and we infiltrated them in tobacco, pepper, spinach, okra, and zucchini. And we've infiltrated them in those plants with the aid of agrobacterium tumefactions. So we started monitoring the infection in local and systemic leaves in a weekly basis, starting from 2 weeks until to 5 weeks post-inoculation. And we collected samples and we ran them into RT-PCR. So from our PCR data, we saw that the RRV infectious clone were successfully able to establish local and systemic infection in all of these inoculated plants. However, the systemic spread was slower comparing to the sub-inoculated plants. As you can see here from those 2 red arrows, the systemic infection started as early as 2 weeks post-inoculation, only in the sub-inoculated plants on the very right. While it took 4 to 5 weeks for this for the systemic infection to show up in the plants that's infected by infectious clone. We also looked at those plants under the transmission electron microscope, and we saw some viral-like particles with a diameter size near 220 nanometers, which presumably could be our virus. So the takeaways. So the RRV distribution could only be associated with the symptoms within the infected plants. And the RRV has expanded its niche beyond the bushes and shrubs by infecting herbaceous plants from different families. And RRV does not have remarkable symptoms on herbaceous hosts, as we saw, except for cucumber, which showed some malformed leaves. And RRV infectious clone is capable of causing systemic infection to herbaceous hosts as well. And RRV takes somewhere between 2 to 5 weeks to cause successful systemic infection. And RRV accumulation within infected plants may vary or even decline with the time in some certain plants. So yeah, by this, do I take questions or we continue with my partner?
Well, I have a question with respect to herbaceous host.
Yes.
Okay, they can be infected, but if I have a field of spots that's infected by RRV, is that going to transfer cirrhosis?
We actually haven't run this experiment, so we haven't recovered the virus, this infected sap from those plants and tried them onto roses yet. We've been thinking of that, but that wasn't included in this project. But probably it's maybe a future direction. Yeah, go ahead.
Yeah, regarding the viral distribution, do you have some statistics about the experiment?
Actually, I don't want to touch on this part because my friend, like Regan, will cover this one, this part, like intensively. We've done like lots of study on that part. Yes. Right.
What would be the reason of systemic virus infection before compared to the static infection?
So people online.
So you're asking why the crude sap or contagious sap is more infectious or more effective than the infectious clone. Actually, that could be attributed to the readiness of the virus in the sap itself, because it came from like plants that have prepared a virus in a viable and contagious form. But for the infectious clone, for sure, it takes time for those constructs to be expressed and then to be replicated and then to move from between the cells and between the tissues. So yeah, I think it's more obvious that the infectious sap it's more capable of causing like faster infection compared to the infectious clone. But we don't have exact, like we haven't done like exact comparison side by side to know the main reason behind that.
Another question on that. So do you or anyone work on a sampling of these herbaceous woody plants from the field near to the those plants? Actually, would say that these plants.
Yeah. We have just like recovered the sap, and we've grown those plants in the greenhouse in controlled environment, and we've inoculated them by mechanical manipulation. But we haven't collected from plants that's grown around roses or rose bushes or something like this.
And that would be a difficult thing to do, because the Vector's common name is the rose moth. My knowledge, its only host are roses species. So in nature, moving from a herbaceous plant, what Vector's going to be doing now? Because Phytococcus fructiferum is not. So it's not going to be on a herbaceous plant. And you're not going to be able to go from a rose to a herbaceous plant with Phytococcus because it's a non-host.
Yes.
So, it's an interesting thing, but I don't think that can happen to in fact, I can't think of a way that it would. Sure, there is a way.
So it could be a dead end for the virus to jump into an herbaceous host. There's no way to come back because the mites, are not host for those plants. But it's still like this virus is mechanically transmissible. So there might be some other way for the virus to spread.